Sikit kena maki dah. I'll just open it here, okay? Why my OneDrive folder is empty in here? No, this is not my account. Sekejap. Tetamu. Am I tetamu? Okay, when you look at this is rather straightforward for the first two phases. So the first phases, which is the carbon fixation or carboxylation, meaning that next week when we learn about photorespiration, it's going to be different. You're not going to be using this. Remember, Rubisco has two biochemical modes: carboxylase activity and also the oxygenase activity. The one that you're holding in your hand now is the oxygenase or carboxylase activity. Remind me. Carboxylase activity. And this is the C3 pathway. Mana. Science at work. Advanced physiology. Dia baru nak apa ni? Lama tak buka. Tu kau ada dalam thumb drive. Dia baru nak syncing. OneDrive is not connected. Why? It refused to connect sebab driver tak update. What? Why computer like this? Stop connecting. Kau internet IP yang tak bagi. That's not connecting. File asing tipu lah. Mana ada? No. Is the internet okay for that phone? Should I change the internet now? Yeah. I don't think it's happening with this one. Tak apalah. Okay, so when you look at these two phases, it's quite straightforward. At the end of phase number 2, which is the green one, the number 2 and number 3 reaction, you get the end product of the reduction, which is apa? Hint. That's the sugar precursor. What's the name? Glyceraldehyde 3-phosphate, and also the product, which is plus, and there's a problem. That was Kau cerita tak? Nope.
Let's see. 2, 3.
Apa ni OneDrive? Kau jangan nak epek.
Yang E.
Bersepah-sepah. Bersepah-sepah. Nampaklah. Oh lupa ni touchscreen. Tutup. How to make it full screen? To slide mechanism. Okay, so up to this point, you have learned that the moment sugar precursor, which is G3P, is formed, it needs to make to exit the cycle or to proceed with regeneration. Okay, if it decides to proceed with the regeneration, that is going to follow. Where's the? Oh, this one. This one. That is going to follow step number 4 until step number 12. If you count the number of steps, in order to regenerate, that is even more complicated process. You got 10 steps in it. Banyak kan? Where else you get 10 steps? When you learn respiration later. In fact, some of the enzymes involved in the cellular respiration in the mitochondria is also present in here. For example, the aldolase and also fructo-1,6-bisphosphatase. Okay. So do you need to memorize all of this? Entah. We like to surprise, right? Yeah. The thing that I want to highlight here is from all of these enzymes that you see, these are in just any biochemical reaction is in fact a limiting factor, meaning that if the enzyme is not happy to do the reaction or not in the capacity to do the reaction, no reaction is going to take place. Even though the enzyme is not directly involved in the sugar formation itself, right? So if you count this, in order to get one sugar precursor, this already 10, 11, 12, 13. You need 13 steps. So in any steps, if something gets coming and disturbed the biochem reaction here, the sugar is going to be interrupted. You're not going to get your sugar. Yeah. People look at you now, oh, good, you get into the university and so on. Do people ask how many steps that you have gone through in order for you to arrive here at the university? Or you don't go to any step, you just suddenly wake up one day already in physio class. Is that what happened? So it's a journey. Dah terkeluar daripada frame. Tak ada siapa nak beli komputer kat aku ke? My computer kan. Acer memang 3 tahun. 3 years out. Okay, next one. Right. If you can see, I kind of highlight this list of enzyme. This enzyme you can find in certain of these reactions. These enzymes are actually somewhat needs to be activated before it can be used. So who's going to activate it? Light. Okay. I know that we have learned about the light reaction, and at the end of light reaction, you get the product of the light reaction, which is the reducing molecule, NADPH, and also the energy molecule, ATP. But guess what? Light has another function actually. It involves in the activation of a number of enzymes. So how does it work? This is what we're going to look at now. How light actually activate these molecule. Let's see. Do I remember this? How many enzymes are there? So there should be Rubisco. There should be 1,6 bis phosphatase. This should be tell me the first letter. S. Pseudohapulose? No. It's hapulose. Hapulose 1,7 bis phosphatase. Number 4, that should be, give me the first letter. P. Really P? P. P. Pollen. Dia macam sebenarnya aku nak lupa, tak ingat tau. Phosphoribulokinase. Betul tak? First letter? N. N. Nangka. Oh, I know. NADP. Glyceraldehyde. 3-phosphate. Dehydrogenase. Betul? So, it means kau tak ada sebab tak ingat, sebab aku boleh ingat. Habislah. Okay. I just want you to understand, in terms of direct activation by the light energy, by the degree of light, is actually this thing. 1, 2, 3, kind of like in the middle, this thing as well. Not really Rubisco. Rubisco actually it's not like a direct activation, but due to a number of things happening due to the highlight. Okay, so go to the next slide. Let's talk about Rubisco. So Rubisco, in order to activate it, there needs to be the presence of a number of chemical situation, right? So if you look at your notes there, Rubisco, without anything happening to it, it's in the form of inactive. Inactive Rubisco. Okay, so we call it E. It's just the enzyme. But the moment it becomes active, Rubisco, we call it C. C what? What does it see there? C, ACM? So this referring to the Rubisco, which is enzyme, ECM. C referring C referring to what? C is actually referring to the activator of Rubisco. In this case, is actually CO2. M referring to the magnesium. Okay, right. Ada tak dalam tu? No, carbamate, in order for the carbamate to happen, situations need to happen first. So these situations is actually the presence of CO2 itself. CO2 that you learned so far is the substrate of the reaction, right? If you read somewhere in the notes, I'm sure it says somewhere that CO2 itself is the activator of the enzyme. Magnesium will come later to stabilize it. Okay, so in this whole new form of activated, it's called carbamylation. It's actually the addition of some functional group to the Rubisco. So why is it difficult to get activated? Can it just do the job? Why it needs to be activated? The thing is, the sugar that you learned this morning, the 5-carbon sugar, ribulose bisphosphate, and another kind of sugar, xylulose. I think the name is xylulose. It should be xylulose. When it is present around the Rubisco, it stops the Rubisco from being active. This is where it comes a bit difficult to understand this thing. The substrate and the reactant is the very thing that stops Rubisco from functioning. Okay, that's why carbamylation needs to happen first, because when carbamylation happens, it will cause the Rubisco to be in the vicinity of CO2 and RUBP, not in the hatred scenario. If it's not in agreement, it will be inhibited. Okay, so if you look at this diagram here, it's got the diagram here. So this is Rubisco, which is inactive. Then comes the CO2. Then comes the magnesium. This magnesium actually comes from the pumping of the thylakoid. You see the thylakoid will at one point has lots of magnesium in it. So it keeps pumping out all this magnesium to the stroma. So there is an increase of magnesium. Why? Because it wants to increase the pH. Okay, and magnesium is a good pH increaser. So it becomes the Rubisco active form. However, this thing can quickly become inactive again with the presence of this protein. This is actually sugar, arabinitol 1-phosphate. Why the sugar keep on interfering with Rubisco? Because sugar, what's the function of sugar? Energy. What else? If you think about it, sugar is sticky, right? It keeps on going to the places it shouldn't go. So this thing kind of getting in the way when the time has not come, right? And plant cell has this mechanism to tell you can come and menyibuk when the time has come, right? So this is the whole reason you are getting this complicated nature of Rubisco. So coming back to here, so Rubisco can become inactive again. However, this can be reversed by Rubisco activase, right? So this Rubisco activase, what is the function? Rubisco activase, with the help of ATP, remove the unneeded sugar from the Rubisco. What sugar? RUBP and XUBP. Is it xylose?
Yes.
Xylose, right? Yep. So you can see now that Rubisco activase is not the only thing that can activate Rubisco. No. The function of Rubisco activase is actually to remove the sugar and to reverse this effect of CA1 protein. However, if the CO2 and magnesium is not present, Rubisco is still not getting activated, right? So fulfill the requirement first. You have Rubisco, the Rubisco is inactive. Why is it inactive? There's a sugar attached to it. Why is it inactive? You got the CA1 protein attached to it. These need to be removed first. Remove this first with the help of Rubisco activase. Once this has been removed, the Rubisco now in the purified state and ready. The moment it is in this state, now it can start to, it will decide now, is the pH correct? That's where the magnesium comes in. Rubisco cannot work if the vicinity is acidic. It needs to be a bit alkaline. Okay, so remove the unwanted sugar first. It will need the sugar, but not now. I need the sugar when two things has been met. CO2 is around surrounding me to do the carbamylation, and magnesium is surrounding me to increase the pH. These two things has been met, then it can proceed to the catalytic cycle. When this happen, it has entered this, then only the CO2 and RUBP come in to do the regular carbon fixation. Okay? Get it? Or you're not following? You got this enzyme. This enzyme is very, very fussy. It needs to be in activated state. However, the very thing that is going to work very soon is the very thing that will stop it from working. The function of Rubisco is to fuse RUBP with CO2, right? However, these two things, actually one of them, the RUBP, make it not functioning because pH is not right. CO2 has not joined with it to form the carbamylation. So fulfill this condition of Rubisco, gets carbamylated, high pH, then only the RUBP come, CO2 come, you'll get your activation of Rubisco. Okay. So can it be just be the Rubisco attached to CO2 without any sugar attached to it? Actually, if you remember from this morning, I said to you, right, the CO2 is not the only thing competing to fuse with Rubisco. What else is competing? Oxygen. Okay. If the pH is not right, oxygen will prevail. Okay. The number will be more. When the number of oxygen is more, less and less of CO2 has the probability to land on the binding site. Okay. How can this be prevented? By increasing the pH, by lowering the temperature. Okay. So when this happens, Rubisco will be open to be activated and then receive both the sugar and also the CO2. Right? Okay? Right. Don't worry, you got the notes. You can read later. Habis lah. Okay, now comes this thing. What's this thing? Ferredoxin thio what? Is it thioredoxin? Ferredoxin-thioredoxin. If you remember, this ferredoxin, you've seen this before. Is it? Yeah, the Z skin at the end of it, right? This is actually not really attached to the membrane, kind of like just right at the end. This thing receives the electrons. It gets reduced. It can do two things actually. It either can pass it to the final acceptor, which is NADPH. That's when you get your reducing molecule, or it can decide is to pass it to another electron acceptor. It's an electron. Okay, differentiate between electron acceptor and electron carrier. So it another ADPH at thioredoxin with the help of the enzyme in the middle here, thioredoxin. Okay, you need this guy in order to do the enzymatic action. The paradoxin containing, okay, actually has a special disulphide two bond. How do you know this disulphide bond? This thing. S two disulphide bond, and this disulphide bond can be reduced, meaning that it receives hydrogen. Okay, it receives from who? From ferredoxin. When it receives this, it will become the reduced form of thioredoxin, and guess what? It can eventually dump all this hydrogen electron to this one of these enzyme. Remember, okay, all of these enzyme are in the stroma. So the point of having thioredoxin to donate electron to these enzymes so that it can be activated. If this enzyme is not activated, it cannot do all of these biochemical steps. Not possible, right? So why does it say that this needs light? Who's driving the movement of this electron? Light reaction. So the more light that you have, the faster the electron transport going to be, the more of this enzyme will be reduced. Okay, more and more of it. This is unlike Rubisco. Okay, Rubisco, even though it needs a Rubisco activase, that some people say that it still needs light, but that is indirect. You don't see Rubisco here. No, Rubisco is not here. Rubisco do not use thioredoxin system to get activated. Rubisco use this special carbamylation with the help of Rubisco activase, right? So the moment it is dark, it will release the pressure of electron movement. When the electron has stopped from moving, can all of this molecule get reduced? No more electron. So hydrogen has stopped moving now. So these enzymes will revert back to inactive state and the Calvin cycle will stop from happening. Right? As easy as that. Right. Is that easy? Very easy. So the story that I've said just now is actually all everything. That is the summary version of it. What's happening. So the thing that you need to know, it all started. You need to go back to the light reaction. Remember this little diagram, the chlorophyll? It gets excited. It undergo the Z scheme. Go up, go down, go up, go down, and then come to the ferredoxin. But this time, it is not ended at NADPH. It goes to the other side. It goes to who? Ferredoxin to thioredoxin. Okay, with the help of which enzyme? This enzyme, thioredoxin reductase. The name reductase should already tell you this is a reduction process. Somebody is donating electron to somebody. Okay, right? So when these all get activated, then this enzyme can start to do all these regenerative properties. From all of these three enzymes, these actually in the regeneration. This is in the regeneration. Okay. This number 5 in which phase? Should be number 2, reduction. This is number 1, carboxylation. 1 is needed for the carboxylation phase. 3 enzyme needs for the regeneration phase, and another 1 enzyme needs for the reduction phase. Right. So no matter where you run, you still need light to somehow activate this thing. Right. So let's briefly talk about how magnesium come about. So you see, when the light is intense, the intensity of light is increasing. What happened to the proton pump? Is it become slower or faster? Slower or faster? The proton. Light coming down the Z scheme. What happened to the proton from the stroma side? Is it going to stay there or is it going to get into somewhere? So it will, the faster the electron move, the more hydrogen H plus will be transferred into the thylakoid. When this happens, what happens to the pH of the stroma? Is it going to increase or decrease? That's going to increase. Okay, so you have fulfilled one requirement of this. To make it more certain, the magnesium which is inside the thylakoid is further pumped into the stroma. Right? Because magnesium, not only that it has the effect to increase pH, it also actually binds to the Rubisco to stabilize it. Remember, Rubisco has undergone carbamylation, right? It needs to be stabilized, increasing the right. So perhaps maybe later, if you want to formulate some kind of foliar fertilizer, maybe it's not a good idea to make it acidic, because you are putting a stop to the Calvin cycle, right? Think about it. Now that you know this, maybe I should create some kind of foliar that is high in, I don't know, magnesium, right? And make it alkaline a bit to assist this activation of Rubisco, right? Okay. All right. It's very, very easy. Oh, what's this? Light control. Oh, this thing. The supramolecular. So this is the final thing for this lesson. So this is about the supramolecular complexes. Oh god, this thing. You know, I'm just telling you the summary story of it. You need to read it anyway when you go back. Okay. So what's this? What's that? What's that? It's actually still the story of these guys, this light activated enzyme. You see, this enzyme, in addition to be needed to be activated by thioredoxin, another thing also needs to happen, because they are sticky. They like to merenyam bersama-sama. You got your phosphoriboglucanase. Betul lah. Nampak gambar mengenyam tu. So you got your phosphoriboglucanase, you got your glyceroldehyde 3-phosphate, then you have, what else you got here? You got CP12, some kind of protein, random protein. You see, in the chloroplast, in fact, in the plant cell, there are many protein molecules floating about. Sometimes it doesn't do anything. It's just there. It's just there because macam kau buat karipat kan, kau dah kepit-kepit tu ada lebih-lebih kan, nak buat apa? Apa tapak tepi-tepi. Benda tu. That benda kelim yang lebih-lebih tu. What do you call it? Excess karipat skin. Yes, you got so many excess karipat skin in this cell. Does it do anything? Are you going to goreng the karipat skin excess? So what do you do with it? You're going to stick it together, right, to forming a ball. So that's what happened here. So you kind of mount it. I mean, like, you don't have to do it. They'll because this is in the liquid, they will start to move, menguit closer to each other. Then it will start to form. Hence, you learn the word now. Supramolecular complexes. Supramolecular meaning that 2, 3 or more molecules join together, not necessarily involving covalent bonds. They are sticking together not because of electron sharing. They can just stick together because they are very sticky and they are good mengenyam together. So this form this one structure, complex structure, we call it supramolecular. Why? Actually, if you learn the protein engineering class, supramolecular actually looks there are many types. Very pretty molecule. Very pretty molecule. Sometimes we don't need the pretty, we need the function, right? Macam cantik tapi bodoh. Buat apa? Right. We need it to be function. If the supramolecular complex is present, like in this case up here, you have 3 proteins come fuse together. You got the phosphoriboglucanase, you got CP12, you got glyceroldehyde 3-phosphate. Come together. Can it do the work? It cannot do the work. Kan? Kau macam pergi mengenyam, lepas tu kau berkepit aja. Dah tu bila macam mana aku nak semayang? That's not going to happen. Somebody need to detach from one another first. So that's why you see this enzyme activity here. When it's in the form of complete, this is the word, ternary. Ternary. Sebab ada 3 benda melekat kat situ, supramolecular. When it's in the form of ternary structure, it cannot do the Calvin cycle steps properly. You are pulling me, I'm pulling you, I'm very sticky, aku nak jalan, kau pijak kain aku. Macam just tak boleh. So, nothing gonna happen there, right? But what happens when you give the light? You see here? There's a light here. When you give the light, this disulphide bond here will start to cause it to dissociate, right? When it becomes its own individual again, right? Then phosphoriboglucanase can do the work, glyceroldehyde can do the work with the presence of high light. This is low light, this is high light. What about another example here? So you got your glyceroldehyde dehydrogenase alone. Ini bukan masalahnya ada kawan yang melekat dengan dia. Dia seorang melekat. It just to attach with itself. Until to the point it's forming this, ni mengenyam ni. There's a word for that actually. I forgot the name. There's a name for that. Something polymer, oligomer. Where is it? Ada tak perkataan oligomers? Jumpa lah. Dia tu. In the dark by forming A8, B8 oligomers. Maksudnya dia tiba-tiba berhalusinasi. There is 8 of me. So let's sticking to each other. Macam why? This is actually your karipat skin lah. From 8 excess karipat. can it do the job? Dia just tak boleh tau, sebab dia dah melekat. Lepas tu lama-lama dia jadi berat. Dia jadi berat and it will start to forming another form. Macam supramolecular ni dia form ternary. Macam ni dia forming oligomers. So these are not the right shape for the protein to do the job. All protein in your body, even in your body, it must assume one specific shape in order to do the job. You got your collagen protein, beta pleated form, circular form. There are so many forms of protein. So these form of protein will determine whether you get some organs, whether you are going to still look pretty and not sagging. That's the function. Okay. So what happens with the dust? That happens in the dark. What happens when the light comes? You go up here, then it will start to detach itself from all of these oligomers members, leaving behind as the glyceraldehyde, dihydrogenase 3-phosphate. Right. Okay. So that is the example. So another importance of the light. It gives a reality slap to the protein. Like stop mengenyam together, do the work. Tadi you learn light is used to activate the thioredoxin, right? Then actually I didn't tell you, but I'm very sure dalam notes tu ada. The way this thing happens is dekat hujung ni, this enzyme has residue amino acid like cysteine. So this cysteine residue, the moment they receive hydrogen, dia kena cleaved. What's cleaved in Malay? Dia kena parangkan. Is that the right word, parangkan? No. Belah. Dia kena belah. So bila dia kena belah ni, dia tak tangkup, sebab tu dia boleh buat kerja dia. Kan? Macam kau duduk melekat aja kat atas sofa tu, bangkit. Aku berapa dah ni? You need to be cleaved from your couch potato sofa. Right? So I think there's a story somewhere in here. So that's how it done. Okay? That's the action of thioredoxin to do the cleaving by the light. This light here just simply to remove the stickiness of the molecule. Right? So they are floating about, then they can do the job. What about all this protein here? CP12, right? You got all this unneeded protein. What they do? They just go to the corner of the cells, like the curry puff skin, and then, if your mom remember, they will reuse. Kalau dia dah siap goreng, dia ambil buang je. So that what happens in the cell as well. If the cell remember, they will reuse again. If they don't remember, macam lebih dah pula, buang je lah. Yeah. So when they decide to throw away this extra protein, that comes the autophagy, mitophagy, chlorophagy, right? Eating up oneself, right? Okay, itu je. Ada quiz? Kau jawab. Tolong. You want to answer number 1? Actually, you will give this later tak? Sorry, number 1. The Calvin-Benson cycle consists of which phases? Cepat ambil. Mana satu? You know what? I'll give you 5 minutes. Try to answer. Betul aku tengok. Dapat jawab tak? Habislah kau. Nanti go toilet kejap. Very easy. Okay, let's quickly check. So number 1, the Calvin-Benson cycle consists of which 3 phases?
Oxidation, reduction, regeneration.
A, B, C? B. Carboxylation, reduction, and regeneration. Agree? Agree. Okay. Tandakan diri sendiri ataupun kawan sebelah kalau dia tak nak. Number 2, the catalytic abilities of Rubisco are activated by. Kau baru je belajar benda ni. Which one? A, B, C, D. Boleh tanda lebih ya? A. Is it correct? Betul. B? C and D? No. In fact, C inhibit because it's a sugar. What's the other one? Xylose. Okay, phosphorylation? No ATP needed in the carboxylation phase, right? That's another phase. So the answer is A and B. Number 3, light influences the activity and expression of key enzyme in the Calvin cycle. In which of the following ways? Number 1, photoreceptor influence the expression of enzyme. Is that correct? Oh, this is good. Mesti macam ramai yang salah benda ni. This is good. This is good. Light influences the activity expression of key enzyme. Okay, actually you just learned with me just now. Alright. A, photoreceptor influences expression of enzyme. Is that correct? Yes. How? Expression of enzyme. Okay, we go to B. Tak apa. KIV. How about B? Ferredoxin-thioredoxin system. Correct. Okay. B correct. What about C? Regulates the catalytic activity of Rubisco. You just learn kat depan tu. You just learn. Kenapa dah ketik ni? Okay, look at here. Any light? So, kenapa kau cakap Rubisco? So, C betul tak? Tak betul. Okay, D, formation of supramolecular complexes of chloroplast system. Betul? Betul lah, baru je tengok buku sebelah ni. What about ion movements? Think hard. What about ion movement? Why? Kalau yes, why? Magnesium aja. Hydrogen. The movement of hydrogen tu. When the light reactions becoming faster, more and more hydrogen get pumped into the thylakoid, leaving B in the stroma, less hydrogen. Therefore, pH up or down? Up. That's ion movement. So apa yang betul? A? Betul? KIV. Tak ada KIV. Masa nak habis dah. The photoreceptor influence expression of enzyme. What is photoreceptors? Okay. Expression of enzyme. Iyalah, betul lah. Dah cakap first light kan? Baru je belajar light activate benda ni. So betul lah. So A betul, B betul, C salah. C betul salah? C salah, D betul. E betul. So satu salah. Okay. Number 4. What is the product generated by Calvin cycle? Alah, tak tahu saja sangat dah. Satu. A, B, C, D. Kenapa tak boleh B? 3-phosphoglycerate. Where do you find 3-phosphoglycerate? Kat mana? Carbon fixation at the end of it. Kan? Now we're talking about at the end of reduction. And finally, ini tak ingat saja. Pagi tadi baru aku tunjuk. Number 5. In order to assimilate molecule CO2, how many molecule ATP required? Ya ke? Why? Okay. So got it right. Okay. Right, datang kelas dah lah. Hi. Okay. So that's about it for the Calvin cycle. Ini baru 10.1 tau. If you go to the front of the page, kau tengok ada berapa banyak 10 kat depan tu. Macam apa lagi lah nak merepek nombor 2 sama nombor 8 tu. Point tu, nombor 1 pun dah macam nak mati. Relax. Bertenang. Okay, any question? Is that clear? Can you do the revision? please do the revision within 24 hours. Okay, that's the only way for you to have a long lasting. Kau yang aku ingat macam mana? Dah belajar sekali tu, 24 jam aku memang paksa diri untuk baca, itu je. Dia tak ada cara lain. Really, dia tak ada cara lain. Itu je cara dia. Engkau orang Afrika ke, orang Eskimo ke, kan, orang have ke, sama je cara. Right. If you don't do it, you are the curry puff skin. Are you the curry puff skin? Right. Okay. So, no question. So, I think we'll just mention now quickly about, we're not going to do experiment treatment this week. Ada siapa nak cakap? Sebab aku tak arah nak cakap. So, someone please rub the board. No. Can I have a kopi? Oh, ada kopi di sini. Kenapa tak warna? Okay, yang aku tak marah dia. Dia memang kedekut sikit. Padahal kan printer tu warna betul-betul tumpah dia. Kering je lah warna. Why do you think I make all the notes colourful? Why do you think? At least it improves about 8% of cognitive ability. Sebab colour excite certain cells, root cell, cone cell dalam mata, that otherwise wouldn't happen if you go monochrome. So you want the neurons to fire while you are reading, so that you remember the colour has been invoked now. Sebab tu. So, beli Canon. Okay. Apa kata ni tak tepi saja? Sebab nak bawa letak barang eksperimen. Ni boleh tutup je. Apa tak tutup? Oh. Apa tak tutup? Dah. Tutup je. Dia boleh terus. So you have your, remember your rice this morning? How does it look? Happy, unhappy? Sad? What did you do this morning? You spray? What did you spray? What is it? Okay. No, I just want you to remember what you have done. Cepat tulis apa benda kau bagi. Ada 3 benda. The name of the thing. Apa benda dalam tu? Go. Cepat. Engkau buat apa? Suruh dia. Benda yang senang dia laju nak jawab. Nak jawab tunggu. Your time will come. Your time will come. How many days now? The 46. 46, that's right. Date? What's the date today? 2011? 20? What happened to 1 until 19? Okay, so this morning you put MPK green. Tulislah polia. Missing. What is missing? Kan. TE. How many TE are there in there? Lupa. Okay, then you got your exogenous silica. Ada apa dalam tu? Silicates. Ada apa lagi? Calcium. Potassium. Lepas tu ada sodium. Then, what to say? Apa benda ni? Dia ada 2 functions. Adjuvant plus pest control. So what to say ni, what is it actually? durian juice, orange juice, orange oil. Okay. I forgot to ni, tapi tak apalah. According to the original plan, we are going to do the spraying of Sara only. Okay. However, when you look at your plant, if you notice your plant today, the leaf has some kind of diseased symptom. Did you notice that? Benda. So this is your homework for you to go back untuk cari. Dia bentuk diamond. Lepas tu kadang-kadang tengah tu ada mata. Brown. Diamond shape. So, apa nama dia? Karat. Karah. Okay.
Blast.
Due to
Fungus.
fungus.
Kalau karah fungus.
Apa lagi simptom yang ada? So this is why when you are dealing with crop, mata tu nak kena buka. Bukan hati perasaan kau je yang nak kena buka. Mata tu luahkan. Apa lagi? Is there any problem? Seeds? What about the seeds?
Bukan potential liver, memang
Apa?
ada seed-borne disease, chemical gas and blood.
Oh, so that's confirmed lah. Huh? Is that confirmed? You know, rice got so many diseases. Super difficult. Actually, rice, I think even without the disease, you can save lots of harvest. But due to these diseases, due to the fungus, due to the bacteria, you know, blight, all these things, greatly diminish the yield potential. All right. So with this activity that we are doing, we want to prove a point actually. First, you can grow rice in urban style, right? You just grow rice in the your wet market tray, and you only apply the granule fertilizer only one time, right? What did you apply? Benda?
ZF.
Yelah, benda. You need to know this. Kau nak let report kau seorang-seorang. Tak ada yang kau boleh nak tinggal jiran kau. Find out that. Okay. Even though CRF is claimed to have long lifespan of up to 6 months, we know that usually this is not the case. Okay, because the thing about this technology is the warmer it gets, the shorter the lifespan of the fertilizer is. Not only that you get warmth, you apa lagi kau dapat kat ladang tu? You get rain, kan? Apa lagi kau dapat? You get wind. Semua benda lah kau dapat. You get disease. So much thing. So what if we change the treatment by ensuring first the plant receive enough nutrition in the form of foliar. Remember, okay, plants can receive nutritional uptake from two doors. The one that you usually learn is from the roots. That's the major nutrient. But the leaves can also receive the nutrient because of what? What's with the leaf that will enable nutrient to enter? Stoma. Tu je. What else? What else? So that's stoma. What else? On the top of the leaf, you got your cuticle, right? So this cuticle is actually not one continuous film. It's actually in the form of jigsaw puzzle. Okay, so when it's in the form of jigsaw puzzle, you will get this crack So we name it cuticular crack. Nutrient that is applied using foliar technology can actually be absorbed into the leaf via stomatal opening as well as cuticular crack. In fact, some technology, even without these two routes, if they use a very expensive adjuvant, the nutrient can actually absorb because it's kind of digest the waxy cuticle, right? But we don't do that because we don't want because usually that is a practice in horticulture. There's no direct sunlight and so on, right? Very shady, LED light, right? So we don't do that, but we infuse our formulation with water seed just now, right? So lupa nak suruh kau print ni, tapi tak apa lah. Yep. So for the next week, I think we need to apply our treatment already because you have started to see these symptoms here. We want to see, can these symptoms be alleviated by providing with the treatment, and can it also assist in the yield production? Can you get higher yield? So you got three trays, right? How many plants you got in each tray? 7. How many? 40 hari tak ingat lagi. How many? 32? 32 bags. Ya ke? Ya ke? Macam 32 ke kurang? So we just assume 32 bags. Okay. So let's put it this way. So the tray number 1, let it be controlled. And we got treatment 1 and treatment 2. Okay. For the control, it will receive the first week, it receive MPK green. Second week, it receive whatever that you give just now, MPK plus calcium plus water seed. A third week, all of them will still receive NPK plus calcium plus water seed, but we need to give treatment to this. What are the treatments? So the treatments is we got 2 things here. Number 1 is we got this thing called phosphoron. So this thing is high in copper. Copper is known to fight the fungal attack. So you can read more about it. Benda yang kau bagi ni. Inilah cerita yang dia jumpa kat kau ni. Dia yang cari ya. Don't blame me. So what can it do? Compete with bad friends. Bad microbes. Apa aku baca? Mana? Oh, terbalik. Compete with bad friends. Phosphoron function liquid formula copper phosphide. So copper is actually by default needed by the plant, but when you give a bit more, the concept is it will strengthen the immunity signals. Yeah. So hopefully, when abundance of copper is sufficient enough, it will help the plant to be strong enough to withstand all this fungal attack. If it's due to the fungal attack, okay, right? But is it going to work or not? That is the question. So this is the content of this product. So we got N13%, P2O5 29.5%, and copper 5%. So T1 would be your phosphoron. How much we going to put? I think we're going to decide later, right? Where else can copper be used as a pest control? Where else? Snail. Yes, you got problem with snail, right? You can apply copper as well. Mana lagi? Bad neighbors, throw at the neighbor. Kan? So that dia jadi baik. You know, in the past, the doorknob is actually made from brass. This thing. You know brass? I don't know apa benda bahasa Melayunya. Brass. Huh? Gangsa bronze. Dia tak tahu. Look up by yourself. I know just now it's called brass. Okay, so this brass is actually an alloy, whatever the name is later you want to find out. And alloy in the past, people kind of put copper in it. Copper in it. So especially in the hospital, whenever people touch the knob to open it, they are also disinfecting the hand because that is a natural germicidal activity there. Right. Kalau kau takut aku scam, you can use the Google to look it up. Huh? Loyang. Loyang bukan kuih yang jual kat Sedang tu ke? Loyang. Brass. But it's an alloy lah. It's an alloy. Okay. So, meaning that it's also good to combat the germs, which is not good for your plant and also for your skin. So, I think it is still being practiced. People are actually touching all these things to disinfect the hand. Okay. Janganlah kau nak disinfect hidung kau pergi so macam tu. Right. Okay. But that's good to know. Alright. Okay. What about the second treatment? Okay. Siapa nak cerita yang ni? Malas sebab aku tak ajar kau lagi kan. What's this? Tada! Did you learn this in soil science? I should be teaching soil science actually. This is actually kind of mycorrhiza. It's you see in this microbes concoction, there's a number of bacteria and also fungus there, which is not a bad thing because you got this in your gut as well. Okay, so these things, if you go to very fertile lands, it's very abundant. You go to somewhere where the tiger lives, right? If you go, why I don't have visa? Let's go to Pahang. You go to National Forest in Pahang, very deep in the jungle. The soil is actually very fertile. Once it's due to the tiger lah, because it has undergone this long, very super long relationship between the microbes, the flora, the fauna, and so on. Did you know that our rainforest can create its own clouds during the dry spell? These plants can spew up aerosols to the sky. When they do this, kind of like cloud seeding program done by the government, program pembenihan awan. So they spew up this aerosol, and it will start to cause clouds to form just above the forest. So they get free shower. So this relationship have been going on for quite some time, not only at the canopy level above it, but also in the soil. So people actually study that. What makes the plant so resistant? Do you go to the Pahang? Who's going to spray malathion right in the middle? Elok pokok tu. Kan? Kau tak bagi baju, sihat je dia. How come? Nombor satu, kau tak ada situ. Kalau rumah kau kat situ, dia tak jadi. Right. So they study the soil. It turns out that a number of microbes or microbe family is actually present in high abundance. When they are in higher proportion, they kind of like suppressing the bad guys. kind of like the microbes in your gut. You know, if you're drinking biotigen, that's a beneficial bacteria, right? If you are eating tempeh, that's actually a good fungus, right? But you try to eat lauk kerui kawin 4 days ago. Especially the one that have bubble coming out of it. Yeah. The one that you're about, should we cucuk now? Is that a good bacteria or not? So these bacteria, whether you like it or not, is always present in your gut. However, the reason that you are healthy now, the reason that kau tak gila lagi sekarang, tak meroyan semua tu, it's because of the healthy proportion of good microbes over the bad microbes. You know your brain is being controlled by the microbes. There's a special nerve, it's called the vagus nerve. Kau lupa kan cerita? Kena cerita. So vagus nerve, look it up. Vagus nerve. It's kind of connected from your gut all the way to the back of your spine. Bukan. Ni. Vagus nerve. Vagus nerve. So the concept here that we're going to apply here is, we have this commercial product which is freeze dried. So there's a tons of this thing. You got Bacillus. Baca. Apa ada?
Bacillus subtilis.
Ya ke subtilis?
Bacillus megatherium. And then Trichoderma ferrite.
Sama ke tak ni?
Sama.
Oh sama. Habis kan tak baca kat kertas. Ambil balik. So Bacillus subtilis, Bacillus megatherium, Trichoderma viridis or Cornigii, or Pseudomonas putida. I don't think putida tu dia ada. Ada ke? Tak ada kan? Putida ni I think this is a special thing. Okay, these two microbes from different kingdom. 2 of them from the bacteria kingdom, another 2 from the fungus kingdom. Guess what? You got that in your gut as well. So we are going to prepare foliar solution, but this foliar solution will be teamed with living being of this. The way we're going to do this, we're not going to do it today, kita buat minggu depan. We're going to use this and then we're going to replicate the bacteria. You need to wake up first, then you need to replicate it. So meaning that you need to give food to this bacteria. Food in the form of what? It says there, molasses. And also, this is my recipe. Kita bagi dia minyak ikan. Molasses as a source of CHO, carbon, hydrogen, oxygen. Energy for the microbes. Fish oil, fish emulsion as a source of protein for the microbes, because protein is needed to do lots of functions in the microbe. So we're going to brew that. We need to brew that. So we're going to use this equipment to brew it. Itu ozon. Kau nak guna ozon ke untuk bawa juga? So we're going to, I just give you a rough visualization how this is going to work. Attach lah dekat bubbler. Kau tengok tiap hari benda ni. Aduh. Cucuk. In our country kan, dia tak ada commercial this kind of product tau. But I look at my friends dekat America, this product is actually kind of semua orang guna. Yang buat this farming. They call it what? Rotational farming. They got sustainable farming. Yeah. They prepare this solution not only to spray to the ground tau. They spray it to the whole plant. So the idea is the solution that is teeming with these microbes will grow heavily on your plant, therefore fighting with this bacteria fungus on the contact surface. Yeah. So we're going to use nanti minggu depan kita buat ya. So going to use the bubbler. It's in the water. So in the water here, we're going to put this thing. And then we're going to put the molasses, the sugar, and then we're going to put the fish emulsion. We let it brew for quite some time. I think we better brew right in the morning kot. So that petang kita boleh ambil dah. Tak boleh, ini bukan donat kau bagi rehat 40 minit dah boleh goreng. At least 6 hours. Okay, air, good thing. Water, tak boleh guna tap water biasa because they got fluoride and chlorine. This thing kill bacteria. Benda ni bacteria. Janganlah. So guna air apa? Air Zam-Zam. Evian. So we're going to use either RO water or distilled water. Okay, I think we got lots of distilled water kat tepi tu. Ambil je. Right. So we use the distilled water to do it. And how do we know? Sebenarnya kan dia elok macam overnight sebenarnya. Macam overnight. Kena buat the day before. How do we know it's ready? You will get all this froth like your Starbucks coffee. This froth maksudnya dia dah bangun. So we get it, put it in your sprayer this morning. Spray as treatment number 2. I think it is not a problem for this thing to be incorporated into the base folia. It's not a problem. Yeah. So we can do that. Just spray for 2 times. Yep. For 2 times. Once is now. Another time is when the flower has started to appear. Okay. So that will be the treatment. And that's it. Sepatutnya tadi you start to learn how to count the tiller. I forgot to ni. Tak apalah. Nanti next week you can do it. Remember, you need to count the tiller number. So once this has been done, kita nak tengok can the disease be suppressed? And by extension, can you get anything meaningful in terms of yield and quality of the rice? Is it going to do any good? Yeah. I'm not saying that this is the best, okay? You can actually do this by yourself. If they make me teach plant nutrition again, I'll show you how. We can go to Pahang. Kan? Visit the tiger, steal some soil. Apa? Fish emulsion. Mahal tu. Okay, so that is all. Alright? So it's very easy treatment. I hope we can get to the harvest stage. If today is 46, by New Year, how many days already?
80, 87, 80.
Let's see what we can get, right? Okay. If we can get some harvest, then that's good. Usually this plant, since it is a single seeding, it's very fast. Previously, we could harvest 110 days only. Actually, 105 days dah okay, tapi I'll just wait for a bit more. Yeah. Now you can know how rice is made. Not in the factory, but in the netting room. Alright, okay. That's all for today. Any question? All good? Nak ambil gambar? Silakan. Right. Let's do it this way. Aku lah suruh dia orang ni kan. We'll prepare some so that we can use on Monday. But for the sake of learning, we'll do another set. You do another set, but you're not going to use that. We use the one that we have prepared earlier because this thing actually it requires about 40 hours to bubbling properly. Dan kau tak boleh guna bekas ni sebenarnya. Kecil, macam kecil. Lepas tu nak keluar buih nak ke mana? Masuk muka kau. Kan? So we'll prepare ahead some, but you still will learn how to prepare this as well. This is very, if you know how to do this kind, you can go to any land that you like. Let's say that your mak ngah got very fertile land, right? Because she buried the husband there. You want, I want the fertility of my mak lang orchard in my home. You can do it. Go to the orchard. Just ignore the dead uncle. Get some soil. Bring back. use the thing that you're going to learn to replicate whatever Pak Alam Microbe has given. Nampak tak? Aku mengajar kau untuk mencuri kesuburan tanah orang. And this folia not only to be meant to be applied on the ground, you can apply it on the body of the plant itself. Alright? Okay? Alright, that's all. Dah. Ambil gambar, ambil lah. Ni boleh buka ni, buka lah kalau dia nak apa-apa. The soil is not in the right condition, it's just look disoriented just like you. So you need the healthy soil. If you go for the soil lah, if you do the hydroponic semua tak, janganlah kau pergi dekat Itafos punya rumah tanaman tu, dia doing the hydroponic, right? Oh, I want to state the fertility here. Lepas tu kau ambil autopot yang ada kokopit tu. Buat apa? Itu bukan subur sebab tu. Dah ambil kokopit, lepas tu nak replicate ni. Mana ada microbe dalam tu? Aish. Alright, okay. Dah. Tolong kemas.
Transport yang jadi dekat unflooded.
Actually ini adalah homework semua orang. I want to see in the apa benda kau ID. No, I will ask again next week. Show me the proof.
Kalau belati dia ke tepi.
Pergi tengok. But I saw tadi bentuk diamond. Lepas tu dia macam ada mata. Bukan Freemason, dia memang ada lah kat situ.
Dia ngering